Good morning, students. This session for first year biotechnology students, semester 2, lecture number 18, unit 3. Unit 3, that is cell biology. In previous class, we discussed about the endoplasmic reticulum. So now we discuss about the mitochondria. Mitochondria is a cell organelle. It is present in eukaryotes. మైటోకాండ్రియా అనేటటువంటిది ఇది సెల్ ఆర్గనెల్. ఇట్ ఈస్ ప్రెసెంట్ ఇన్ ద యూకారియోట్స్, ఆబ్సెంట్ ఇన్ prokaryotic organism. In Greek, mito means thread, chondria means granular. In Greek, mito means thread, chondria means granular. It is otherwise called as, mitochondria is otherwise called as, the other name is the powerhouse of the cell. So the energy utilization, the energy producing organelle in the form of ATP, that's why it is called as powerhouse of the cell or storehouse of energy. Storehouse of energy. They are granular filamentous organelle. Mitochondria are granular filamentous organelles. Coming to the history, in the year 1850, Kollikar observed in muscle cells, they are described as sarcosomes. In the year 1850, Kollikar observed mitochondria in muscle cells, they are described as sarcosomes. After Kollikar, the next scientist is Fleming. In the year 1882, he described mitochondria are thread-like structures. They are used as a term is known as phyla. So Fleming used mitochondria are thread-like structures. They are called phyla. After Fleming, the next scientist is Altman. Altman termed as mitochondria are bioplast. In bio means life, plast means germ. Other name, according to Altman, they are called bioplast. Bio means life, plast means germ cells. After Altman, other scientist, in the year 1898, the scientist name called as Benda. He developed a staining technique according to the Gram staining technique. By using the crystal violet solution, he developed and he termed mitochondria. The mitochondria name was coined by Benda. During 1850 to 1898, other scientists called as Kollikar called as sarcosomes, Fleming used as phyla, Altman coined the bioplast, and Benda given a perfect name is called mitochondria. The mitochondria term was developed and given by Benda. He used crystal violet stain solution. By using crystal violet, he observed mitochondria. It was termed by Benda. The credit goes to Benda. He used mitochondria for first time. Then after the development is going on, during 1940 to 1950, electron microscope revealed by Ruska. By using here, two scientists were observed. They are Palade and Porter. Palade and Porter given the particular three-dimensional structure of mitochondria. It was first used by Palade and Porter. These two scientists were given a particular shape of mitochondria. Till from that time to till today onwards, mitochondria shape is as like it is a the structure given by the granular or filamentous, or it produce like spherical or oval in shape. సో ఇక్కడ మైటోకాండ్రియాకి ఈ స్ట్రక్చర్ అనేటటువంటిది పర్టికులర్గా మనకి ప్రొడ్యూస్ చేసినటువంటి సైంటిస్ట్ పెలాడే అండ్ పోర్టర్. బై యూజింగ్ దీస్ టూ కైండ్ ఆఫ్ పెలాడే అండ్ పోర్టర్, the given ultra structure of mitochondria by using electron microscope. ఎలక్ట్రాన్ మైక్రోస్కోప్ని యూజ్ చేసిన తర్వాత ఆల్టమన్ అండ్ ఇక్కడ మనకి పెలాడే పోర్టర్ ఈ యొక్క పర్టికులర్ షేప్ ఆఫ్ మైటోకాండ్రియా అనేటటువంటిది ప్రొడ్యూస్ చేయడం జరిగింది. సో దిస్ ఈజ్ అబౌట్ ద హిస్టరీ ఆఫ్ మైటోకాండ్రియా. వన్స్ అగైన్ ఐ రిపీట్, మైటోకాండ్రియా ఈజ్ ద యూకారియోటిక్ సెల్ ఆర్గనెల్. So here in Greek, mito means thread, chondria means granular. So this other name as mitochondria is called as powerhouse of the cell. Their other name is the storehouse of the energy. By energy in the form of ATP. Energy, the units of energy in cells, they are called as adenosine triphosphate. They are granular structures, they are filamentous organelles, they are granular and filamentous organelle. During the history, in the year 1850, Kolliker observed in muscle cells, they are called as sarcosomes. In the year 1882, Fleming observed mitochondria or thread-like structures, they are called as phyla. 1892, Altman used these mitochondria are given the particular name is bioplasts. These bioplasts are life germ cells. And the year 1898, Benda given a perfect name of mitochondria. Benda coined the term mitochondria. He developed a staining technique. He used the staining of mitochondria is nothing but a crystal violet solution. Then the development is going on. 1942-1950, electron microscope was revealed. Then the time Palladio and Potter given a particular shape of mitochondria. This was a granular filamentous spherical in shape. So coming to the size of mitochondria, the mitochondria size is 3 to 4 micrometer and the diameter contains 0.5 to 1 microns in diameter. So the shape of mitochondria is the granular shape, filamentous shape, spherical or oval in shape. So the distribution, the where it is present, mitochondria are present in the eukaryotic cells in the form of cytoplasm. These cytoplasm, it is a viscous or jelly-like substance. So here mitochondria can move, they can flow one side to another side. That means up and down or right to left. During the surrounding by the nucleus, they are rotating according to their particular shapes. So here the distribution is nothing but a cytoplasm, where the cytoplasm is a germ-like or a jelly-like fluid. That kind of substance mitochondria can move from one place to another place itself. So coming to the structure of mitochondria, here mitochondria structure contain two membranes are present. That is called, first one is the outer membrane. So it is the protection of the mitochondria, it having the outer membrane. The second one is the inner membrane. Inner membrane means this inner membrane is nothing but a plasma membrane. Look like it is a thin-like structure. Here the mitochondria can folded like forms. Here mitochondria are folded like forms. These folded like forms to produce a cristae. That substance is nothing but cristae. Here some particles are present. They are called F1 particles. These F1 particles to bind the granular structures, they are called as ATP. Here an enzyme is present, that is nothing but ATPase. ATPase enzyme can produce here some type of energy containing storehouse of the cells. They are form ATP. So next, here some matrix is present. The matrix where it is located, inner part of the mitochondria, folded like form of mitochondria. So the matrix is present. Then here some particles are present. These particles are called as F1 particles. So the folded form of inner membrane is nothing but a cristae. Then here some droplet vesicles are present. They are called granules. It becomes, so coming to the particles, the large size of vesicles are nothing but a granule. So by according to the granule, it given by chondria. Then it have a genetic material, nothing but circular DNA is present. Where it is located, it is present in the inner folded like form of mitochondria structure itself. So it is about the structural difference. So here the structure contain two membranes. First one is the outer membrane, second one is the inner membrane. So in outer membrane, it gives a particular shape of the cell. It is present in the outside. Then here the outer membrane and inner membrane in middle periplasmic space is present. So here the inner membrane is a folded like form of plasma, invaginated plasma membrane. These foldeds are nothing but a cristae. The folded particles of inner membrane is called cristae. Here some particles are present. These particles are called as F1 particles. Here the energy can produce in the form of ATP by using an enzyme is called ATPase. Then going to the functions of mitochondria, the mitochondria functions are very numerable, uncountable. So here it is mainly useful in the respiration process. By using here aerobic respiration is present. By means presence of oxygen levels, the respire can utilize by these mitochondria. Here the respiration is called aerobic respiration, presence of oxygen, whereas anaerobic in the presence of carbon dioxide, that respiration is called as anaerobic respiration. Here the aerobic respiration can occur. So all metabolic pathways are produced by these enzymes. It can perform different 60 type of enzymes can produce like oxidative phosphorylase and carboxylase. Next here some enzymes are present. They are called 60 different type of enzymes. They are useful in metabolic pathways. That metabolism is called as intermediate metabolism. Then it they can provide food for energy. It gives food for energy utilization. Then it perform oxidation phosphorylation. It presents in the form of Krebs cycle. That means here Hans Krebs introduced tricarboxylic acid cycle. Then that carboxylic cycle, they can utilize some enzymes. These all enzymes produced by the mitochondria. Then the chemical composition of mitochondria having here lipids, RNA, trace amount of DNA and some proteins are present. The composition contains 25 to 30% of lipid content and 5 to 7% of RNA, little bit the trace amount of DNA, and the final amount of proteins are 60 to 70% of proteins are present in that kind of chemical composition. In chemical composition, lipid content is present, RNA is present, DNA is present and proteins. So lipid content 25 to 30%, RNA 5 to 7%, DNA trace amounts, proteins are 60 to 70%. Here can you observe the high amount of chemical composition having 60 to 70% of proteins. The proteins are high amount, RNA, DNAs are low level, and second category contain lipids. So the mitochondria, it gives energy to the cell. Which type of cell? It contain all eukaryotic cells. All eukaryotic cells, here plant cells are present, animal cells are present. So the all the type of cells, here energy utilization by performing the mitochondria, the energy in the form of ATP. That ATP is nothing but adenosine triphosphate. These ATPs are useful for the metabolic pathways. So by using oxidative phosphorylation and aerobic respiration, and it provide food for energy, and it produce more than 60 type of different enzymes are producing. So this is about the mitochondria. So ఈ మైటోకాండ్రియా అనేది మీకు ఎస్ఐ క్వశ్చన్కి అడగొచ్చు సెల్ ఆర్గనెల్స్ నుంచి, లేదా షార్ట్ ఆన్సర్కి అయినా వస్తుందండి. దీనికి సంబంధించిన నోట్స్ అనేది మీ వాట్సాప్ గ్రూప్లో నేను అప్లోడ్ చేయడం జరుగుతుంది. సో అలాగే మీకు సెకండ్ యూనిట్కి సంబంధించి కూడా మీకు కొన్ని లెసన్స్ అనేటటువంటివి మైక్రోబయాలజీ లెసన్స్ అనేటటువంటివి జరుగుతూ ఉన్నాయి. సో రికార్డ్ అనేది కూడా మీకు మీ వాట్సాప్ గ్రూప్లో అప్లోడ్ చేయడం జరిగింది. రికార్డ్ దగ్గరికి వస్తే ఆల్రెడీ మీరు ఫస్ట్ సెమిస్టర్ రికార్డ్ రాశారు, దానికి కాలేజ్ రికార్డ్ తీసుకుంటే కంటిన్యూషన్ చేయండి. సర్టిఫికెట్ ఉంటుంది, అక్కడి నుంచి కంటిన్యూషన్ చేయండి. ఇన్ కేస్ కొంతమంది ఏం చేస్తారంటే రికార్డ్స్ బయట నుంచి తీసుకున్నారు మీరు, అలాంటి వాళ్ళు ఏం చేస్తారంటే అది షార్ట్ రికార్డ్ కాబట్టి అగైన్ యూ కెన్ పర్చేస్ అనదర్ రికార్డ్ ఫర్ సెకండ్ సెమిస్టర్. సో సెకండ్ సెమిస్టర్కి మీరు వేరే ఇంకొక రికార్డ్ తీసుకుని దాంట్లో మీరు రికార్డ్ రాయొచ్చండి. సో రికార్డ్ పోర్షన్ రాయొచ్చు, నోట్స్ అనేది కూడా మీరు వాట్సాప్ గ్రూప్లో ఇస్తున్నాం. నెక్స్ట్ మెయిన్ మీరు ఫోకసింగ్ అనేది థియరీ మీద ఫోకసింగ్ చేయండి. సో థియరీలో మీకు ఆల్రెడీ మనం Unit-3లో ఉన్నాం. Unit-3లో మైటోకాండ్రియా, ద నెక్స్ట్ టాపిక్ ఈజ్ రైబోజోమ్స్. నేను నెక్స్ట్ వచ్చే టాపిక్ మీకు రైబోజోమ్ గురించి చెప్తాను. మీకు ఏమైనా క్వరీస్ ఆర్ డౌట్స్ అనుకుంటే యూ కెన్ కాంటాక్ట్ మీ. ఓకే, థ్యాంక్ యూ స్టూడెంట్స్. హ్యావ్ ఎ నైస్ డే.